Okay, thank you very much, Tilak and President and organizers for asking me to give this talk. Okay, so firstly, what I'll do is I'll talk about an overview of biologics, a quick overview of biologics, because that has come into internal medicine and sort of specialty medicine in a big way in different areas of medicine. And what we know is that medicine today, we have a population-based, one-size-fits-all model of care. And what we're trying to get to is a more personalized medicine, predictive, preventive, patient-centric model. We are using genomics plus other important, having a foundation of clinical sort of basic clinical medicine and clinical findings, and then using genomics to be able to target these different immune pathways and genetic pathways in different areas of medicine. So basically what we're trying to do is we're trying to get medications that are not toxic and beneficial, because rather than giving every patient the same sort of medication, which sometimes can be toxic and not very beneficial, we just want to target the specific group of patients so that we get optimal benefit from that particular group of drugs. So what I'll do in this next 10 minutes or 15 minutes is talk to you about the basics, monoclonal antibodies, about biologics and conventional drugs, and biosimilars to round it off. Now we know the immune system, viruses, bacteria, and fungi can attack our body. You have the immune system, the defense system, which consists of antibodies, white cells, and complement. And these have to work in concert and at appropriate levels to be able to keep the sort of harmful agents outside our body and not causing damage. Sometimes what happens is these components can be defective, and then you have immune deficiency, and we have to boost the person's immune system. At times, you find that the immune system, that's antibodies and white cells, can attack our own body cells, and this is called autoimmunity. And there are different autoimmune states, which will be discussed in the next few lectures. Sometimes you find that the immune system attacks innocuous substance in the environment, and that is allergy. So you have a normal state of the immune system, you have underactive immune system, and then you have overactivity in a dysregulated fashion, in an abnormal fashion. And in those instances, we have to give sort of immunosuppressive agents or other agents, including biologics, to bring it back to baseline. So that is basically what we would do when a person with immune-related issues comes to see us, across from an immunology perspective or in organ-based specialists or at internal medicine level. So we know that there are different cells that are present in the body. From the bone marrow, it originates with cells, white cells, neutrophils, eosinophils, lymphocytes, T cells, B cells, etc. The B cells go direct into the bloodstream, the T cells go into the thymus, get educated there, and the non-harmful T cells come out of the thymus. Different types of T cells, you have CD4 cells, CD8 cells, B cells, which have CD19 on the surface, and NK cells, which have CD56. When we look at the T cells, we have different types, Th1, Th2, Th17, and Tregs. Now, depending on the condition, different cells have a greater role to play. Say Th2 cells would have a greater role to play in asthma and allergic disorders. Th1 cells would have a greater role to, and Th17 cells would have a greater role to play in autoimmune diseases, which will also be discussed in the symposium. And the important thing with regards to the immune, so you find that the B cells produce immunoglobulins and through plasma cells, and the immunoglobulins have these different components, which all of you would have learned in medical school, the variable component and the constant component. Now, it's important that the T cells, B cells, and the antigen-presenting cells work in concert together with the cytokines produced by the T cells. The cytokines are just chemicals that the cells use to communicate with each other to be able to carry out the immune function. Once the immune system gets activated, you have to bring it down into baseline again, because otherwise you would find that the physiological functions are not met, and that is what happened in COVID, where you found more cytokines produced and a hypercytokine state coming. So this is a schematic to show the interaction between a T cell and the antigen-presenting cell, APC. You find that the T cell receptor is there, so you have an accelerator of the process, and that would get the immune system activated. But once the immune system gets activated, you do need to have two breaks. One is the CTLA4 break with CD18, and the other one is PD1, PDL1, and this is the basis of immunotherapy, the discovery of which won the Nobel Prize last year, or 2 years ago. So the important thing is, just because you have the immune system gets activated, you have to have a break, the CTLA4 break or the PD1, PDL1 break, or other breaks to bring it back to baseline. And in certain autoimmune diseases, this is important. In certain cancers, this is important. So next, going on to monoclonal antibody. That was a quick outline of the basics of the immune system and the different breaks and the accelerators of the immune system. Quite some time ago, it was Milstein and Kohler got the Nobel Prize for finding the technique for producing monoclonal antibodies. That is of one type, not polyclonal, but one type, monoclonal. And based on the different monoclonal antibodies and how it is produced, you can name it, and this is the sort of schematic that they use for naming the different monoclonal biologics that we have. It's MOMAB if it is mouse, it's ZMAB if it's chimeric, if it's humanized, it's ZUMAB, and it's UMAB if it's human. So you're looking at the name, we can sort of get an idea from what source this monoclonal antibody been derived. And so if you look at biologics, you have the novel biologics, including the antibodies, monoclonal antibodies, and proteins, growth hormones, interleukin, etc., and including vaccines and some peptides. And then you have the follow-on biologics, which are biosimilars and the biobetters. Biosimilars and monoclonal antibodies, I think biologics, I'd be just having a slide going on. So how do you compare the biologics with conventional drugs? It's like a sort of the analogy that is being given is, if aspirin is like a bicycle, it is like a sort of a large biologic, it's something like a business jet, because the complexity is important when it comes to biologics, and that is very important in the manufacturing process, together with sort of the regulatory components in ensuring that the biosimilars, etc., are similar to the biologics that are being used. So what are the sort of comparison between traditional biologics? You have multiple effects, short-acting, oral administration, quite a few of those are intravenous, and then the interactions, but biologic specific, long-acting. So you're trying to be more specific and take out different components of the immune system, or try to stimulate different components of the immune system to have its more specific effects, rather than going blunderbuss, like sort of trying to affect all parts of the immune system in one go. You want to be a bit more specific. Biologics have been quite prominent in recent years. Many FDA approvals have been biologics in recent years. This slide shows that schematic over the sort of years, but the problem is cost. Can we afford it? And this was a sort of editorial some time ago, but that is an important thing, because as more biologics come into play, can we afford this different drug? And where does the world, who should we get to target this drug? Because we should target them appropriately so that healthcare resources are used appropriately. So finally, the last few slides is about biosimilars, because I spoke to you about biologics, what is it, including monoclonal antibodies. I told you about the conventional drugs, and talking about biosimilars. So if you look at the schematic, you have the traditional medicines, you have generic medicines, which are easy to produce because it is not so complex as opposed to biologics. Then you have biologic medicines, large molecules, and then you have biosimilars, which there are some characteristics that are laid down for it to be a biosimilar, similar to structure, pharmacodynamics, pharmacokinetics, etc., and safety and efficacy. So this is the sort of schematic that we use. There are the originals, the true innovator, and I've given some examples here. Then you have the biobetters, the biosimilars. It becomes a bit more affordable over a range of clinical settings. And and then you have the non-original biologics and the biobetters to improve the efficacy and safety of administration, and those are the biobetters. And certain companies who produce the original innovator also get some biobetter to get better characteristics of the drug. So traditional generic, 2 to 3 years, biosimilars take some time. I mean, just because a biosimilar doesn't mean that it won't take some time. There is some development cost, and then you have the different biologics, which are the first generation and the second generation, which are the biobetters to improve characteristics of that group of drugs. So trying to compare biologics and biosimilars, you find that these are the sort of characteristics. I won't go into great detail because of time constraint, but immediately you can see that there are differences between the biologics and the biosimilars that are present, that are given here. This has been for some time. Yemen Commission sort of published draft guidelines on market and approval of biosimilar in the UK, and over the years, several biosimilars have come into use within the NHS and in different sort of settings. FDA approved the first biosimilar in 2015, and this was a sort of a biosimilar timeline. This is only up to 2018, but going on 2021, we have a number of biosimilars, including adalimumab, just come as a biosimilar. So I've just given a list of the drugs, the biosimilars that have been authorized over the different years here, and this is the next slide, which again shows a similar pattern of a number of biosimilars together with the biologics. There are parent biologics that have been authorized. So valuation of global demand for biosimilars has been rising rapidly. 2020 level is very much higher than this, and it's a big market across the world for different pharmaceutical companies. So what I've done in this quick update was told you about the basics of the immune system, told you about the T cells, the B cells, and those acting together in concert, monoclonal antibodies, Cole and Milstein, and how they were produced, how they are named to depend on the source that it's produced, biologics and conventional drugs. I compared those two, told you that conventional drugs take shorter and the properties of the conventional drugs, and then told you some aspects about biosimilars and how it compares with biologics, and what are the regulatory, there are some regulatory components with regards to biosimilars to be able to use those in different, and to come into clinical practice and to be able to use. Thank you.